I'm coming to you live and direct via satellites from Oxford, England. And I'm here at ONI Laboratories in, as I said, Oxford. So I'm here because we have purchased the only microscope on the planet where you can rapidly see an exosome and importantly see the surface markers of the exosome. The problem that we've encountered is that in the exosome field, you go as the state of the art progresses. When I first started, and I'll go through this in the talk, we use protein concentration, and I really wanted to see this exosome. Previously, we could only see it by electron microscopy. But to see it by electron microscopy takes weeks. You have to try different types of metals, you have to fix the thing. It's not something that you can do rapidly. So eventually this microscope called a nanocyte came out. And I was extremely excited when that did come out and we could purchase one. It's about a $150,000 microscope, but what it did was it bounced light off of the particle. Now, knowing that I had a protein concentration, I had done protein sequencing and messenger RNA sequencing, I knew I had an exosome because I could see, the contents of it. But I couldn't really see the exosome. I was just going sort of on space. So when I got the nanocyte and I could see the particle, I assumed those were exosomes. Now, the machine has an ability, it has a laser on it, so I should be able to see the surface of the exosome. The problem is these particles are moving around and you can't really see them and they're not strong enough. So because we've been doing this for such a long time, I was approached by this company, it's Oxford Nano Imaging, and they told me about their microscope. And when I show you these images, you're going to be sort of astounded or flabbergasted. So this thing has a resolution of 20 nanometers and you can actually see the particle. Now it's not so easy to use, which is why we're over here getting trained on it. But it's really going to change the way we define an exosome. I don't believe that you can use the word particle anymore to define an exosome. So if you took, for instance, amniotic fluid, you throw it on there, there's so much stuff in amniotic fluid. There's meconium, there's vernix, all these things make little particles. Proteins can even clump together sometimes and make what looks like an exosome. So, we have a lot of competitors that are out there saying it's 300 billion, 70 billion, whatever. All that's going away. If you don't have this microscope, and there's only 50 of them in the world, there's only 25 in the US. We purchased one 5 days before the National Institutes of Health. Only people like AstraZeneca have one. You're not really going to be able to say that you have an exosome. So let's start my presentation. Okay. So this is what we're really talking about today, being able to take wounds and make them heal more rapidly. This is a friend of mine. He fell off a bike, so we were able to get him exosomes within 2 days. He placed them topically and inside of the wound. And if you look at him 8 days later, you really can't see much. And that's really what we see. We speed up healing. And we do that two ways. We have both growth factors in the milieu, but we also really stop inflammation. And here's an interesting anecdote, is that there was a paper recently that put together a wound healing model in a mouse. They put the same wound on both mice. They injected one wound topically and peripherally, and then the other wound, they injected the mice IV. And interestingly, the IV mouse healed more rapidly than the one that was applied peripherally and locally. And why is that? Because inflammation is a systemic thing, right? I mean, you might have a wound on your arm, but your lymph node is up in your armpit. So if you don't stop the inflammation everywhere, or even in the spleen, you really aren't stopping inflammation. So that's very interesting. Here's another girl who we actually got to meet last week. This is a patient of Dr. Chernoff in Indianapolis. She woke her dog up by mistake, and her dog bit her lip off. So this is 7 days later after injecting it peripherally into the wound and topically. So it's really amazing. And the girl's about to get married. She was there with her fiance. It was really a sweet situation getting to meet her. So as I said, my name is Duncan Ross. I'm the founder of Kimera Labs, and we are the owners of Exosomes.com. So we have been doing this since 2012, and really it's been a progression, a very long progression. It started about 20 years ago when my father got myelodysplastic syndrome, and he was a scientist. I'd been accepted to UN Medical School before this, and I'd gone off to Europe to work on the internet. And he got MDS, and so we started researching it like research scientists would, reading all kinds of journals such as Blood and the Biology of Bone and Marrow Transplantation. Unfortunately, he died when I got into graduate school, but it was really because of him. He was the impetus that I did join the biochemistry department. And after spending 3 or 4 years in the biochemistry department, I switched to the immunology department, where I got to actually study cord blood transplantation as we were trying to get for him. So the concepts of bone marrow transplantation have been around since the 50s and really have led what we do. This is Kimera Labs right now. We have 6,000 square feet over on the left and 21,000 square feet over on the right. So we have quite a bit of space. We have 2 clean rooms right now. We'll probably have 4 in Q2 of 2020. And we're going to start doing a number of open houses there where we get deeper into the biochemistry and science that we're going to discuss today, let you play with some cells, let you play with the new microscope, and really understand what we're doing. So if you want to come visit us in the new year, please do so. This is the structure of Kimera Labs right now. There's somewhere between 40 and 50 employees if you take consultants into account. Every one of these bigger circles is an MD or a PhD, so we have quite a number of people working, and we do quite a lot of quality control. We're also working very diligently on our FDA RMAT applications. We have 2 INDs that we're working on, one for craniofacial neuralgia and one for keloid scarring, which you can talk to Dr. Sanders about. This is some images of the new lab. We have a lot of space there on the left. That's where we're going to be doing our trainings. A lot of offices for people, very nice laboratories. And, you know, we're very excited about that. This is an example of one of our clean rooms here to the left. You know, people, one thing you have to consider is I've been making this product since 2014. I didn't have any quality control. The cost that I have charged people, I still have people that basically pay costs because they've been helping me since the very beginning. But my cost keeps increasing. So, you know, at what point do I have to up the price to them? Because you know, you can't just make an exosome for $50 a vial. I mean, you could take amniotic fluid and put it in a vial and say it's exosomes, but that's not really exosomes. So what we're doing is very expensive, and we are doing that so that we can create a reproducible product that can be FDA approved. And we need to have that done by 2020. So this is the model that I used to work with in the immunology department. It's a model of graft versus host disease. And it was a good model because it taught me very easily. I could see fibrosis and the death of the mice, but I could also see that if I used a regulatory T cell, I could stop that inflammation. So that kind of gave me the idea that cellular factors or cells could stop inflammation and possibly treat disease. So one of the molecules that was most important in that kind of suppression of graft versus host disease or suppression of inflammation is TGF-beta3. I'm going to talk a lot today about TGF-beta3 because it's a protein that if you put it in a dish with a CD4 T cell that's activated, it will turn that T cell into a regulatory T cell and turn it off. And that is a permanent conversion. And that's extremely exciting that we know this. And so if we could manipulate this or handle this protein, can you imagine what we could do? When I was in graduate school in the biochemistry department, I used to produce recombinant proteins. One weekend, I figured out that what I'd done in a weekend was worth $2 million if I could express one specific protein. So luckily, there is a cell that can do that for us, and that's really what I'm doing. I'm using it as a farm and trying to get to proteins like that, as well as others. So since I was using regulatory T cells, that's what my lab was concentrating on, I was aware that there were other people using mesenchymal stem cells to suppress graft versus host disease. In these clinical trials here, they would use 14 million mesenchymal stem cells to suppress chronic graft versus host disease. That gives you a number and a basis because chronic GVHD is a very, very inflammatory system, right? So you can think of this MSC then as a golden goose. If you look over at the anti-inflammatory factors there, you have IL-10, TGF-beta3, and PDGE. Those are all secreted by regulatory T cells. Regulatory T cells really are the kings of the inflammation world, except that they're really hard to culture. Mesenchymal stem cells are a lot easier, but mesenchymal stem cells have an added benefit that regulatory T cells don't have. They have hepatocyte growth factor and other types of growth factors that can cause growth. So what you should think of from now on is that a mesenchymal stem cell is the center of your body, the same way you used to think that your heart was the center of your body. This cell is controlling the growth of the cells around it. You know, if you're in a liver, you know, or in a heart, one cell doesn't care about the cells growing around it. Something has to orchestrate this in the entire organism. And the way people think that this works is that you have this mesenchymal stem cell sitting there on the vasculature, secreting paracrine factors, secreting those anti-inflammatory factors and those growth factors. If you think about yourself now, you're not stricken down with arthritis, right? If you weren't still secreting anti-inflammatory factors, you would be. The thing is, you're not growing. Certainly, you know that. You haven't been growing since you were about 20. So this cell we're looking at here has turned off its growth program that secretes these growth factors and is only now secreting anti-inflammatory factors and keeping you in an anti-inflammatory state until there's inflammation. And to cause inflammation, you need multiple steps. You've heard of this two-hit theory of inflammation. So, you know, most of the cells in our body secrete anti-inflammatory factors to keep us like that. So in 2012, I thought I was going to help providers like yourselves create stromal vascular fraction or bone marrow stem cell transplants, quote unquote, even though we're talking about really mesenchymal stem cell transplants. So I filed for an IRB for the use of stromal vascular fraction in COPD. And I had some very good outcomes. I could take people's FEV1, which they would usually come in around 0.8. They would sound terrible, you know, and most of the time they didn't have a lot of funds because they couldn't work. And I could usually increase that to about 1.4 or even more, sometimes 1.8. And that would really change these people's lives. They could go out and, you know, work on their cars, you know, they could go travel. Some of them could even fly in airplanes again. Our slogan used to be fly again. Because, you know, you're at 8,000 feet in an airplane and a lot of people can't fly if they have COPD. So the issue was that generally these patients would relapse 6 to 8 months later. Now, if you've just charged people $13,000 and they don't have a lot of money to begin with, you can't do that on a repeated basis. It's not ethical. So I started trying to look for different ways to help them. I was aware of that Mesoblast clinical trial I just showed you where they used 14 million cells. So I decided, well, let me shoot for a target of 7 million mesenchymal stem cells, because in stromal vascular fraction, there's only 150,000 mesenchymal stem cells. So if I could grow each patient's cells up to 7 million, maybe I'd have a better effect. And let's see what happens. I thought this because we had known that if you add more cells to a wound healing model, the wound heals more rapidly. So it follows that more cells are going to be more beneficial. Why did I use autologous cells instead of allogeneic cells like cord blood, which has been a big thing in the past couple of years? Well, the thing is, cord blood has always been a 351 product, meaning you have to have a BLA license. Only transplant centers would do that kind of thing. Remember, I'm from a transplant background. I was really flabbergasted when I saw that people use cord blood in orthopedics. Cord blood is only licensed for use in leukemia transplantation. That's the only reason we bank cells. Unfortunately, the FDA posted something, or put out a guidance that said, as you see on the top, that hematopoietic stem cells derived from peripheral or umbilical cord blood could be used as a 361 product. But they meant autologous. They meant your own. They meant that if you have multiple myeloma, you'll go through an autologous transplantation, and the banking of those cells and reinfusion of to you was legal. But they didn't state it correctly. And that's something that we're going to get to later. The way the FDA states what they want is important. It's a legal document, right? And we have to follow that. So another reason I didn't want to use third-party cells is that mesenchymal stem cells are immune evasive. They are not immune privileged. People call them immune evasive because they only have MHC class 1 on their cell surface, HLA type 1. If you put them into another animal, they'll be rejected in a period of about 24 hours if put into the circulation. That's a lot faster than you would think. So that's really why I never wanted to use third-party cells. This is what happens if you take third-party cells and put it into an animal. So on the top here, you can see that this is a twin, a syngeneic mouse. So 3 days later, you can see cells that are put into that mouse. If you use an allogeneic mouse, you can see that 3 days later, all the cells are gone. So that's what happens when you take somebody else's cord blood and put it into you. So for those reasons, I wanted to avoid that and try to grow the patient's own cells up to, like I said, 7 million cells. So I started doing that, and I started having some really nice outcomes. Now, I started to run into trouble, though, once in a while. Generally, I would take out stromal vascular fraction, and I could grow it in this very expensive, it's called a xeno-free chemically defined medium. It's 10 times more expensive than if I used human serum or cow serum. There's nothing in it besides chemicals, growth factors rather. So I'm using this medium, which at the time, you know, we really didn't have a lot, and I'm trying to grow the cells of the wife of the plastic surgeon that I was actually doing this stromal vascular fraction studies with. And I couldn't get her cells to grow. She had COPD. But I had a paraplegic patient whose cells were growing just fine. So I was starting to give up on these cells over here, and I said to myself, all right, look, I'm going to throw these out. Why don't I just take the supernatant from the 18-year-old, the liquid, and put it on the 70-year-old and see what happens. And right away, the 70-year-old cells, because I was looking at them so carefully I could see this, started growing. And that's when I had sort of my eureka moment that I've been throwing out the good stuff all this time. The paracrine effect is real. The proteins are in there. And having worked in the biochemistry department, I know exactly how to manipulate proteins. So let me start making a product out of the proteins that we see in the milieu, in what's called the supernatant. So there were some studies. I mean, I wasn't completely surprised by this. There were some studies from the 50s where a young mouse was attached to an old mouse and the circulation was mixed. And the young mouse got older, but the old mouse got younger. The old mouse even improved its ability to run a maze. It's interesting, though, that the young mouse got older. So we have to talk about that. What's happening is that these exosomes that were present in that supernatant are going from the young mouse to the old mouse. I mean, mesenchymal stem cells are not moving back and forth. Those stem cells are attached to the vasculature. They're not going anywhere. But the proteins can go back and forth. The old mouse actually also has some exosomes. They have these senescent vesicles that are very big, and they can cause cells to die. So that's why the young mouse got older, because the old mouse is sending those. So you can start to think of different possibilities. There's a couple of people using leukapheresis to try to take out half of the serum of older patients, and that therefore takes out a lot of these senescent bodies. And they say they're seeing nice results. I haven't seen it, but I'm actually planning on trying it and using exosomes to, our young exosomes to supplement that. So the advantages of cells, as we have said, is that the persistence is limited to days. It also takes 6 weeks to 6 months to try to grow your own cells, and that's not going to help us very much with burns. Third-party cells will also lead to rejection, as we saw, and allogeneic cells are not FDA approved, and that is in fact why the FDA has been going around and shutting down all these cord blood manufacturers. We have been FDA inspected. Our FDA inspection was from 2017 and was just closed a month ago. It was closed because we're not doing anything wrong. We don't use cells, all we use is cell factors, and we'll talk about the way the FDA regulates that soon. And the real problem is that cells are old. If I get cells from a patient that's 50 years old, they're not making growth factors anymore. They're really kind of useless. And you may have seen this in your own attempts at regenerative medicine. So it had been shown that this complete medium that we're talking about, the supernatant, was able to work as well as MSCs in a model of emphysema and many other models. So this was starting to make me really believe in being able to just use the secretions of the cell, the exosomes. So, but the dosage that I started using was based on the number of cells that I had seen work in my IRB-approved trials and my uses of expanded cells. So I already knew how many cells I needed to use for something to work. So I just went and started using the same amount of secretion. I didn't know how to count exosomes at that time, and neither did anybody else in the world. So I came out with a number that was based on a therapeutic number that I had gotten from our treatments. So when I found out that I could use just the secretions, just the exosomes, that was in March of 2014. In October of 2014, I was at a party and a friend of mine threw water on a gasoline fire and burned his face. He was very, very upset, and he's black. And the next day he's crying, he's thinking he's going to be disfigured for the rest of his life. And he said, "What can you do to help me?" I said, "Well, I have these factors in my freezer that I think can cure you, and I would like to apply them to you 3 times a day." Well, he asked some other physicians, and you remember at that time, PRP was sort of all the rage. And the plastic surgeon friend of his said, "Well, I'm going to take your blood and I'm going to create PRP and you can put that on there." Now, that would have been the worst thing to happen. He would have scarred because PRP causes inflammation no matter what you do. And it does that because there's mitochondrial DNA in the PRP that it looks like bacterial DNA and causes inflammation. That's the purpose of PRP. So I sent her a number of papers because, you know, there was literature in Asia and Singapore to back up what I was saying. And she allowed me to go ahead and do what I wanted to do, which was apply our exosomes 3 times a day to his face for 7 days. And here you can see what he looks like 7 days later. And on the right, you see him 6 months later. Now here's a zoomed in version of that. What you're seeing here is new skin being made by young exosomes. And these young exosomes make everything that baby exosomes make. And if you look at a baby's skin, it's very soft and supple. And indeed, that's what you see. So the difference here is that by using young factors, we can make young skin, we can make young hearts, we can make young knees, we can do whatever we need to do. So this is what an exosome actually looks like. And as I said, you could previously only see them by electron microscopy. They're about 100 nanometers in size. And what they do is they protect their contents. And their contents are made up of messenger RNA and proteins. It's very important because if you take proteins out and put them on a table, they're gone in minutes. First of all, they denature. Second of all, the environment is full of proteases that will cut them up. Messenger RNA is not much better. Messenger RNA has RNases in the environment that will break them up. So it's really important that we protect these things as they're moving around throughout the body. This is what, if I just said that they're 100 nanometers, well, exosomes vary in size. Remember, exosome science is very novel. That's one of the reasons I'm in Oxford right now. I need a machine that can look at the exosome. I do not need to know just what the volume of the exosome is or what the volume of, or what the size of a particle is. If you come in and you say, we have more exosomes than Kimera, well, on the left, you have a 70 nanometer bone marrow exosome. On the right, you have a 137 nanometer Kimera exosome. And in fact, these are both particles by what we knew before. The thing is, if you account that they are both exosomes, the Kimera exosome has 8 times the volume. So you don't need to have 2 or 3 times as many red exosomes as Kimera. You need to have 8 times more than Kimera in order to state that you have more factors or more messenger RNA. And as these companies haven't even done protein sequencing or messenger RNA sequencing of their products, you know, you really don't know what you're talking about. So Doug Spiel, who's probably there with you right now, has coined this phrase that exosomes are the penicillin of our generation. Well, I just want to add to that and add the word young. And that word young is so important. When I say 3, you say young, okay? 1, 2, 3, young. I hope you did that, even though I'm not there. But it's key because I've given this talk so many times and people come out and say, okay, so exosomes, but they don't get, why can't I just get exosomes from my patient's fat? That's not what I just told you. I just told you that young exosomes are the key. And that's what Kimera Labs focuses on. So this is a nice image that came out around 2011-12. So people have been talking about this for a while. Exosomes are how cells communicate. They don't just throw proteins out into the environment where they'll break down. They throw them out covered by exosomes. And there have been these ISEV extracellular vesicle meetings since 2012. I have gone to every meeting of theirs since sometime in 2017. They have local meetings as well. And that's where real exosome science exists. That's where academics present information. That's where we try to function. Generally, the exosome science that you hear on the street is basically taking what we say and saying that you have more. But there are ways to qualify exosomes, and there are people working on very interesting things. They're working on cancer exosomes, obviously wound healing exosomes, manipulated exosomes. It's a very exciting field. And if you're really that interested and think you can follow science, you can go to these meetings. So where can you get exosomes from? It is true that exosomes are available in every one of these fluids. There's exosomes in blood, but they would be old if you just got them from an adult. Even if he's 20 years old, he's still old. He's not growing anymore, right? You can get exosomes from urine. And in fact, we're starting a whole diagnostics division because of this microscope. And we're going to try to look for it, for instance, prostate diagnostics, et cetera, in urine and picking out the correct exosome, which is really hard. And then diagnosing what's inside that exosome, looking at the messenger RNA and the protein contents of that. You can get exosomes from amniotic fluid. It's true, they're young. Kimera Labs sells amniotic fluid. I love it. I take the vernix out of it, I sterile filter it, and I have a product which definitely has growth factors in it. And I've qualified that and quantified it. Now, if I put it on a nanocyte, it says that there's 300 billion particles in there. But do I have 300 billion times the number of growth factors as the mesenchymal stem cell particles? No, I do not. What that tells me is that you don't have 300 billion exosomes in amniotic fluid. you have 300 billion little dots of junk. And the reason I don't isolate exosomes from amniotic fluid is both because the exosomes from amniotic fluid or the particles come from epithelial cells from the mother, but because I can't. I cannot separate the good stuff from all the junk that's in amniotic fluid. So I don't bother. I work with a specific cell line where I know what I can get. And that's really how drug companies do it. If you look at bone marrow, you have exosomes in there, sure, but that's old. And where do you get bone marrow? Who donates bone marrow? When you were in college, they asked you to donate blood, but nobody asked you to donate bone marrow. They asked you to enter a registry in case somebody needed a bone marrow transplant, but they didn't take bone marrow from you. In fact, the place where you get bone marrow donation is the same place where you get kidney or heart donation, from cadavers. So if somebody says they have bone marrow exosomes, there's a good chance they spun down that bone marrow and are using the supernatant. Milk has exosomes in it, human or otherwise. So here's an interesting thought. When you drink cow milk, you take in orally cow exosomes. And you express those cow exosome messenger RNA for up to a period of a month. That is, in fact, why dairy farmers are so big. They're all drinking unpasteurized milk, and they're very large. An interesting anecdote I heard is that the zip codes that can really pass the Army's entry test are all the same zip codes where people have dairy farms and drink unpasteurized milk. So there is a benefit to that. Cell cultured conditioned medium may have exosomes in it, and in fact, that's how we do it. So every cell makes exosomes, including tumor cells. If I were you, remember I need these tumor cell exosomes for diagnostics. If I were you, I would not tell your patients that you're using exosomes. I would say I'm using placental-derived growth factors. Because when they go and they Google exosomes, the first thing they find are tumor exosomes. The thing is, tumor exosomes are much, much bigger. I showed you the volume difference between a 70 nanometer and 130 nanometer particle before. Exosomes from tumors are somewhere between 600 and 800 nanometers. They're very, very big. And that's so they can encapsulate all the DNA that they need. So the benefit of exosomes is that they can be sterile filtered. We produce these inside of ISO 7 rooms, inside of ISO 5 isolators. They are sterile filtered right there inside that isolator and put into a vial. So the chance of contaminating that is very low. But just to be safe, we test them for sterility and endotoxin before we release them. Another benefit is that they can be concentrated. We have a number of different concentrations of exosomes, and we can make as many different types as we want. They will also not be rejected by a third party because these are from mesenchymal stem cells. If I was taking them from a dendritic cell, they might have class 1 and class 2 on their surface. And in fact, I might want to make an inflammatory exosome in the future for use in tumor therapy. However, what we're using right now has very little, if any, HLA on the surface. Now, the benefit of this microscope I just got is I can stain for that HLA and see it on the surface of that exosome, which was previously impossible. It can also be frozen, and storage is key. The reason that we're so excited about exosomes is because you don't have to store them at cryogenic temperatures. They can be frozen in your regular freezer and kept there for up to 6 months. We keep them at -80 for a period of a year, but they're probably good for a much longer period of time. Another nice thing this microscope is going to do for us, if we just looked at, is it helps us look at the stability of the exosomes. I just yesterday looked at a sample of an exosome that we looked at 3 months ago. We left it at 4 degrees Celsius for a month, and then we looked at it just now on this microscope, and it's completely fallen apart. So you will want to use that exosome in a period of about a day after thawing it. But it's really fun to be able to actually see that. So this is our method and how we do this. We use 1 placenta. And there's people out there saying, well, you know, they use 1 placenta, they've been using it forever, it's old, they grow these cells out. No, that's not how that works. We have 1 placenta, we grow it out for a period of 3 to 4 weeks, and then we freeze down the cells into a large master cell bank. Every month we go and pull 1 aliquot out of that and start growing those cells for 4 weeks where we collect the exosomes right here, right? At the end of a month, we throw those cells out. We just toss them. We don't sell them, we don't do anything with them. Then we pull out another aliquot from the freezer and start the whole process over again. In that fashion, we can create a reproducible product. Because while not a lot of people care about that, I have to get FDA approval by November 2020. And I can't do that without a reproducible product. In fact, I don't know how I could do it without this microscope because I can't prove that I'm making the same amount of actual exosomes, not just particles. I could show growth factor panels, I could show messenger RNA, but how was he going to do it? So we sterile filter that conditioned medium. Now, this is where the amniotic fluid exosome people stop. All it is sterile filtered amniotic fluid. But the definition of an exosome by the International Society of Extracellular Vesicles is a purified exosome. And one of the methods used in a lot of literature is ultracentrifugation at 100,000 G. Now, the centrifuge in your clinic is about 1,200 G. So this is a very robust piece of equipment that if it breaks, the rotor can go through the ceiling, and this has happened before. So you spin that overnight at 100,000 G, and you end up getting the exosomes to pellet down, and even proteins. We then take off the culture medium, which is definitely a drug. We toss that, and then we resuspend those exosomes in 0.9% saline. Now, what we end up with is 5 billion exosomes per ml. Now, that is something that was carried over from the number of cells that I saw work. If you've taken any other exosome or any other claim of another particle, it has nothing to do with Kimera's products. In fact, it was probably a misnomer of mine to do that, a mistake of mine to do that, because I could never have known what an exosome is, but I know that 5 cc's of our product works, and that's the key. So we then quarantine and test release that. So the benefit here is that because we can concentrate it, and I know how many cells you need to use over a period of time to get a desired effect, I can make exosomes more effective if we'd like. We could take the amount of exosomes secreted by a cell for a whole month and concentrate that down into one vial. But we do what we find we need to do. And this is going to be a drug, and it does have a therapeutic amount. You don't want to give too much. It's immunosuppressive because, as I'm going to show you, it has that TGF-beta3 in it. So you want to dose patients according to what they are, and that's what physicians do. So this is what's really going on with the science. You have a mesenchymal stem cell secreting exosomes. Those are taken out like trying to throw a football to somebody and caught by a recipient cell that it wants to tell to do something. That messenger RNA that's in there, and yes, there's protein, but the half-life of a VEGF protein is 4 hours. The half-life of a VEGF messenger RNA is 4 days. So the way the body really transplants information is by sending the blueprint for the protein to the area where it's needed and then making thousands of those proteins there. Which here you see the messenger RNA making protein. There are also microRNA in here which can control the expression of this messenger RNA, the translation of these proteins. And those are also very important and something that we're researching. In fact, if you wanted to make a modified exosome, you would manipulate the microRNAs that are available in that exosome. And we will get there. Right now, we have a product that works. We want FDA approval for it. And I have 6 PhDs working for me whose goal is to make the next exosomes. So think of it like football. You've got 2 different teams here. You've got these, if these helmets were exosomes, this exosome throws well and this exosome runs well. Just because they're both exosomes doesn't mean they have anything to do with each other. And one will beat the other in certain circumstances. If it's raining, these guys might win, right? That's how it works. So the key with the young exosomes is that we secrete about 110 proteins at this point. When we're young, we're secreting 300 proteins. Many of those are growth factors. That was one of my techs there in the background. Thank you, Zoe. So there's about 300 growth factors that encompass all types of things, anti-inflammatory, growth instantiating, etc. So how do we validate the exosome? I find it important lately because a lot of people say a lot of things. Well, like I said, since 2014, the only way I could do that was by protein concentration. And this is an example of our instrumentation. So the first thing we do is look at the protein concentration. That tells us that there's protein in there. It doesn't tell us the messenger RNA. It doesn't tell us the number of particles or the number of exosomes. We go by this amount of protein in a product. And we've been repeating that same amount since 2014. And that's why we've come up with these protocols and efficacy strategies. You can look at the surface of exosomes using flow cytometry, but you can't look at individual exosomes. It's kind of hard. These beads will grab a number of exosomes at the same time and try to, increase the signal. It's okay. It's data, and that's what that data looks like. So I know I have exosomes because I have CD63, CD9, and CD81 on their surface. Now, not every exosome has each one of those markers, and that's where exosome science is right now. That's the state of the art. Just saying that you have a particle doesn't mean you have an exosome. Did you have a CD9? Did the CD9 come out of the Golgi apparatus? Do you have CD1? Is that from a lysosome? I mean, that is exosome science, not just I have X number of particles. So, but what matters? What actually pushes the buttons? Proteins push the buttons. So we did this growth factor assay looking at the proteins, and I did this for myself, not for you. I wanted to see what the difference between my amniotic fluid was and my mesenchymal stem cell exosome fluid was. And I might have thought that they might have looked the same. If mesenchymal stem cells were really what was creating the exosomes and amniotic fluid, they would be the same. When I saw this, I immediately realized they're not the same. If you look at the amniotic fluid, the profile looks like a fibroblast profile. In fact, it also looks like a PRP profile. So the interesting part of this is that it looks like an anti-inflammatory PRP. And in our hands, it seems to function that way. But if you wanted to look at the number of exosomes, quote unquote, or the number of particles by nanocyte, you're going to see 300 billion particles. Over on the right, in 1 billion particles of mesenchymal stem cell exosomes, you see growth factors, but they're different. Now, which one do I really care about? I told you I care about TGF-beta3. So here you see in mesenchymal stem cell exosomes, we have 340.5 picograms per ml in this assay of TGF-beta3. But if you look at the 300 billion, quote unquote, exosomes that are really just particles, there's nothing there because those particles are not exosomes, all right? Now, if you look on the left, you'll see somebody's room temperature amnio in a liquid form. The only way you can have a room temperature protein milieu is as if it's freeze-dried, if it's lyophilized. Anybody that walks in with a room temperature liquid product that has proteins in it, there's nothing in there. And somebody challenged me and I ran it at the same time. And as you see 0, 0, 0 going down, if you actually see any signal, that's because the antibody is probably bound in a non-specific fashion. So our preferred method for corroborating placental MSC data, let's say we needed to go from one placenta to the next, is to make sure it secretes the same number of messenger RNA, because messenger RNA are really easy to validate. When you try to get proteins from exosomes, really remember the exosome is protecting the proteins. So if you throw an antibody in there for an ELISA, it can't see what's inside the exosome. So I actually had to make up my own protocol to open the exosome so that I could see those proteins and take 2 different experiments and put the numbers together. So it's not a really reproducible way of doing it. But messenger RNA sequencing is. We can tell you exactly what's in there. We get gigabytes of data. And even if there's a protein that I haven't thought of before, we can go back and look for it. So this is a really reproducible way of doing it. If you think about that young mouse, old mouse experiment that I was looking at, their conclusion in that was that a protein called GDF11 was the one that they correlated to the most use-effective type of protein. Well, we can see that we have it there, and we can also see that we have TGF-beta3. This is the way that I like to think. Messenger RNA data. Okay, so how did we progress from protein concentration to now? When we were able to purchase a nanocyte over 1.5 years ago, we started, really getting what we thought were exosome data, right? This is what you see when you look. In our case, because we have a pure population coming out of a mesenchymal stem cell, those are exosomes, and I'm seeing that now with our new microscope. But I couldn't tell you for sure, right? But if I put water on that machine, we also saw exosomes, but we saw particles, right? So we started to get very nervous, and we wanted to try to prove those CD63, CD81, CD9 that I showed you before by flow cytometry. So, these things don't happen overnight, getting these assays going. And we were starting to get more and more data convincing ourselves that we had what we needed. But, then we started to have competition come saying we have more exosomes, meaning particles, than Kimera. Now, as I said, some of those particles were smaller. We did know from the nanocyte that our exosomes were about 120, 130 nanometers, which is what the rest of the industry shows. When you go to those ISEV meetings in Japan or Barcelona, most people's therapeutic exosomes are that size. So we had more and more confidence that we had what we thought we had. We had protein data, messenger RNA data, now we have size data. All these things tell us, yeah, we have an exosome, right? So, if we could do electron microscopy every day, we could see our exosomes. But you know what? This electron microscopy doesn't tell us where those exosomes come from. We can't see the surface markers on those. So we don't know if it's just a bleb or if it came from, like I said, the endoplasmic reticulum, the Golgi apparatus, or whatnot. So we can't see them on a regular microscope, even with a 100x oil immersion system. So recently, we have had a new type of microscope. And this microscope that we're using is called D-Storm fluorescence microscopy. What you're looking at right here is antibodies to the surface of a mitochondria. And this is a mitochondria moving around in 3D. Now, this is the kind of data the world has been waiting for. This is when you're starting to look at very, very small particles with confidence. without an electron microscope. So if you look at it from my point of view, we create 3 batches a week, and we have to qualify those batches as to what's in them. Now I can go further. All those little things you see shooting around, those are exosomes moving around inside of a cell. I can look at exosome uptake, and this is without even zooming all the way in on the microscope. So now I'm going to show you what the new definition of an exosome is. Because of all this data we have, and because of the industry stating that, you know, X number of particles is more than Kimera, remember, we have a therapeutic dose. And we are now going to state that therapeutic dose in a unit that we're going to call the Ross unit. And that Ross unit is going to be the size of the exosome, the number of exosomes, the surface molecules on that exosome, and we're going to link it to a growth factor or 2 that I haven't decided on yet, and I probably won't tell anybody, but it's going to help us qualify each batch to the next. So this is an actual exosome. This is the first time in the world people have seen this. This is a Kimera exosome. That is, you can see on the surface of that exosome that there's CD63 and CD1 on the surface. Over here, you have an exosome that has a little bit less. This is why we were having so much trouble with the flow cytometry, because we didn't understand this. We were getting this data that was changing, and it was hard for us to understand. When you can look at it, you can very easily understand, oh, this one has less CD63 because it came from a different part of the cell. They're both exosomes, they're both about the same size, but they're different. So the more information we have, the more we start to change our opinion of what an exosome is. And thank God I got this now, because we're taking these 2 applications to the FDA, and this is how I'm going to qualify that we have repeatable batches, and I promise you that they'll accept this. As I said, we bought our instrument 5 days before the NIH, and the only people that really have this instrument right now are larger pharmaceutical companies like AstraZeneca, et cetera. So what's the end goal? We're looking for FDA approval. I learned a while ago from a Nobel laureate named Mario Capecchi that there is a link between the immune system and the brain. So we're working, one of our INDs are with Shelley Jordan from UCLA, and when he's worried about the fact that when you inject exosomes IV, they distribute to the spleen and liver. Well, what if you want to work on neurological disease? Well, he has a method of using focused ultrasound to focus energy and increase blood flow in a specific area of the brain. And we're hoping that we're going to be able to show that exosomes preferentially leave in this area. He's treated about 300 patients this way. This is actually a patient, I brought him. So this boy had cerebral palsy. This is actually one of our distributor's children. And you can see where the neural tracts died when there was a lack of oxygen. Now, interestingly, one of the PhDs we have working for us used to grow placental mesenchymal stem cells for Celgene, but she also grew induced pluripotent stem cells. So we're going to take a cell from this boy, we're going to differentiate it into his own neuron, and then we're going to start to make exosomes and neurons from him. But how are we going to get those there? Well, we're going to focus this ultrasound in that area, which you can buy for $20,000. And what he really wants is he wants for all of you to have one of these in your office. You'll send him a functional MRI like this, and he'll give you the coordinates to target and the amount of power to target that energy to. And this way you'll be able to treat neurological disease in your office. Here you can see that by focusing about 15 seconds of power, you can increase the blood flow by 4 times. So, you know, he has very robust data to say this is going to continue. This is a paper that I can send to any of you. Exosomes are very small things. They diffuse a lot in a solution. You have to use a gel. For now, we suggest using PRP. You all know how to do it. Platelet-rich fibrin matrix is a very good thing to use. The reason that you can do this with our product is because it is isolated. We're hearing reports from competitors that not to use their product with PRP because there have been some episodes of anaphylactic shock. Now, I saw things clumping when I didn't isolate the exosome. That's why I had to start isolating it. And that's really where all of our cost comes from. It costs us 7 to 8 times more than the competitors to produce such a pure product. But what you can see here is that if you inject exosomes alone, you will not cure a chondral defect. But if you include it with a gel, you will. So it's key that you use the exosomes the right way. Let's talk about regulation for a second. I was inspected 2 weeks before this came out in November of 2017, and they were adamant that I tell them what was in my product because they wanted to make sure that I wasn't selling cells. And I was very excited to tell them that the whole point of my company was that you don't need cells. All you need is the extract or the secretions of those cells, known as to us now exosomes. But it says here, secreted body fluids, for example, amniotic fluid, are generally not considered HCTPs. Now, I told them I sold amniotic fluid, and I told them I used it for orthopedic uses, which obviously injecting into a joint. They closed their inspection of me, so I guess there's no issue with that. But what we know is that there are definitely exosomes in amniotic fluid. So what they're saying here is that they are not regulated. If you go down here, you see the following articles are not considered HCTPs. Secreted or extracted human products, such as milk, which has exosomes, collagen, and cell factors, except that semen is considered an HCTP. So how can you tell me now that exosomes are regulated? Now, I fully believe that exosomes will be regulated in November 2020. Are you saying that all I have to do is remove the lipid membrane and that these cell factors will then not be regulated? I mean, I want regulation. We're spending a lot of money on that. It will help us to get rid of the competitors. But this is what they said, and this is what we're functioning at this time. This is a copy of my inspection report. This is from 10/19/2017. She came twice, the first time about the amniotic fluid and the second time about ExoGlow. So you can see that ExoGlow was inspected and thought about by the FDA in 2017. What's interesting is that this inspection was closed on 8/22/2017. So for 2 years, they kept this open and went around to clinics that were using my product, looking for me marketing the product in a way that I shouldn't. But remember, this was developed for burns. This is why I made it. And this is how we advertise it. That physicians go off and use it in these strange ways, like Dr. Jordan there, was never part of the intention. But he's treated about 350 people at this point and has had wonderful outcomes. What you can see here is that there are no FDA 483 inspectional observations. If there had been a problem, I would have gotten a letter that I had to respond to. Now, I don't want to be too proud about this, but what I'm saying is that until November 2020, which they have given us, I have to move as quickly as I can to get an IND application in. And the only thing I'm lacking right now is this qualification of the exosome, which is why I'm here in Oxford. If you want to prove that to yourself, you can look it up. There is a database. You can look up any laboratory that comes to see you. You can see if they're really a laboratory or just a marketing group. Generally, they're just a marketing group. They don't have their own lab. Almost 100% of what we make goes back into the laboratory, whereas 100% of what they make goes back into making nice marketing materials. So try to consider that and try to come visit the lab in Miami. That's pretty much all I have to tell you. One last thing is that a number of things that have come out of these International Society of Extracellular Vesicle meetings is that exosomes are being used to cure cancer, or to stop it at least from growing. And there were 5 labs at this last ISEV meeting talking about that. So how does that work? Well, let's say you have pancreatic cancer. Generally, you have a P53 or a P10 mutation. So that protein is not being expressed. Even if it's a DNA mutation, what's the end result? The protein is not being expressed. So if you put the correct wild type messenger RNA back into a cell using our exosome, for instance, then that normal P10 and P53 can be expressed. Now, I'm not suggesting you do that right now because you would have to know the genetic profile of the tumor, but I'm saying that that is what people are doing in the lab. You can take an umbilical cord exosome, put it in a tube of in a dish with a myeloid tumor, and the tumor will start growing if the correct messenger RNA are there to replace any that have not been expressed. So that's where we're going, and that's why we're moving into diagnostics as well, to see if we can get to a point where we can create patient-specific therapies. So that's all I have for you today. Thank you very much for joining me. Hope you're still awake, and I hope you had a fun day with everyone there. And hope to see you in the lab in Miami sometime.